Hi guys. So today we are talking about chapter 4, which covers cellular metabolism. So metabolism itself, remember from chemistry class, is just the sum of all of the chemical reactions happening in your body. So whenever somebody refers to your metabolism, they're just talking about the chemical reactions happening in your body. If you have a high metabolism, then the chemical reactions are happening faster. If you have a low metabolism or a slow metabolism, then the chemical reactions are happening slower. So cellular metabolism just refers to the sum of all of the chemical reactions that are happening inside of the cell. So metabolic reactions will usually occur in a pathway or a cycle, and we'll talk about those in a little bit. But first off, there are two types of metabolic reactions. Anabolism are synthesis reactions. So you are building something. So you're taking small molecules and putting them together to build a larger molecule. This requires energy. On the other hand, catabolism is when larger molecules are being broken down into smaller ones. And this actually releases energy. So you can see in the picture on the bottom, if you take two monosaccharides and build a disaccharide, that's an anabolic reaction. On the other hand, if you take the disaccharide and break it down into the two monosaccharide components, that is catabolism or a catabolic reaction. So anabolism provides materials for maintenance, cellular growth, and repair, but again, it requires the ATP that are made from catabolic reactions. So an example would be dehydration synthesis. Basically what happens is water is removed, and where the water was, a bond is formed. So smaller molecules are going to be bound together to form larger ones, and water is going to be produced in the process. So you could think of it as those molecules are then missing water. So think about what happens to you when you're dehydrated. When you're dehydrated, you have or don't have enough water. So you have lost water, just like these molecules have. But this is going to be used to produce polysaccharides, proteins, and triglycerides. So again, the picture shows you if you take two monosaccharides, you remove that water molecule and put a bond there and you have disaccharides. This is showing you putting a glycerol molecule with 3 fatty acids to get a triglyceride, and water is given off in the process. If you put 2 amino acids together, you form a peptide bond, and the dipeptide is formed with water given off in the process. So all of these are removing water molecules and creating a bond where the water was removed. Catabolism breaks down larger molecules into smaller ones, and this actually produces ATP, so energy is given off in the process. The example is hydrolysis. Hydro means water, lysis means break. So you're using water to break a bond. So this is used to break down carbohydrates, proteins, lipids. Basically what happens is water is just put in where the bond is at, and it splits the bond. So now you have your monomer components. It's the reverse of dehydration synthesis. So here in the picture, if you have that disaccharide and you add water, it's going to break that bond between the 2 monosaccharides, and you're going to end up with 2 monosaccharides. Same thing with the triglyceride breaking it down and the dipeptide molecule breaking it down. So the rates of these reactions have to be controlled. The breakdown and energy releasing reactions have to occur at rates that balance out with the building up or energy utilizing reactions. So in a nutshell, you have to have enough reactions giving off energy to fuel the ones that require energy. Imbalances in reaction rates can damage or kill a cell. Different types of cells, of course, are going to conduct specialized metabolic processes. All cells in general perform catabolic and anabolic reactions, and enzymes really help control the rates of the catabolic and anabolic reactions. So enzymes are proteins and they're catalysts, meaning that they speed up chemical reactions. Globular proteins catalyze specific reactions, and they're going to increase the rates of those reactions. They do this because they lower the activation energy that is required to start the reactions. So it's like if you imagine you're trying to push a rock up a hill, and it's a really big hill. It's going to take a lot of energy to do that. What an enzyme does is chops the hill in half. So now imagine you're pushing that same rock up a hill half the size. It's not going to take as much energy. So that's what catalysts do. That's what enzymes do. They come in and they lower the amount of energy that's required to start those reactions. The good thing about enzymes is they're not consumed in the reaction, so they can be used over and over and over again. So you do not need a lot of enzyme to perform a function. Each enzyme is going to be specific to its particular substrate, and oftentimes they are named alike. So sugars, for example, usually end in OSE, like lactose, maltose, glucose. The enzymes end in ASE. So most of the time, if you know the name of an enzyme, you can figure out what it does. So lactase, for example, breaks down lactose. Sucrase breaks down sucrose. ATPase breaks down ATP. So you kind of get the idea. So the ability of that enzyme to recognize the substrate is going to depend on the shape of the active site of the enzyme. So basically, the enzyme has an active site, which is where the substrate is going to actually bind. So the active site and the substrate have to match. So it's called a lock and key fit, basically. It's specific enzymes for specific substrates. So, for example, sucrase is not going to break down lactose. So there are certain factors that affect enzyme action and can actually alter the conformation of an enzyme, which the shape of the enzyme is crucial to its functioning, because if it doesn't have the same shape, it's not going to match its substrate. And if it doesn't match its substrate, it can't break it down or catalyze the reaction. So excessive heat can alter the conformation of an enzyme. Radiation, electricity, some chemicals, if the pH is out of the enzyme's range, and some poisons. What will happen is it will cause the enzyme to denature. And denaturation is the inactivation of an enzyme because of an irreversible change in its shape. So what happens is if it gets really hot, the enzyme will actually unfold. We know that proteins have to be folded in order to be functional. So the enzyme will unfold, and now the active site is no longer the same shape, and it can't bind to its substrate. Metabolome is all small molecules are part of the metabolism in a cell, tissue, organ, or organism. So a Human Metabolome database stores vast amounts of information about all of these molecules, which we call metabolites. It's estimated that human cells have at least 2,500 of these metabolites, but we have not really identified even half of them. So it's kind of like they hypothesize that there's 2,500, but we're not sure what they are exactly. Metabolites are being analyzed and added to the database to record the concentrations that are present in different cells under different conditions. So we want to understand how the pathways and enzymes interact with these metabolites, and if there's any drugs or food or anything that can change it as well. So the information is being used in toxicology to try to figure out how people process drugs and discovering new drugs even. And then, of course, diagnosing people, screening newborns, and transplant monitoring, which is a very important thing because transplants are so high risk anyway, and there's such a chance for rejection that we have to be very careful. Okay, so metabolic pathways are just a series of enzyme-controlled reactions that lead to the formation of some product. Each new substrate is the product of the previous reaction. So there's a bunch of steps involved. So you have substrate 1, which will turn into substrate 2. That's catalyzed by enzyme A. Then substrate 2 will turn into substrate 3, catalyzed by enzyme B, and so on until you get the final product. As I said, the names of the enzymes often end in ASE and contain the name of the substrate involved. So lactase breaks down lactose, protease breaks down proteins, lipase breaks down lipids. Each step of the pathway is going to be catalyzed by a different enzyme. A regulatory enzyme is going to catalyze one step of the pathway, and that's kind of going to set the rate for the entire sequence. So it will determine how fast it goes or how slow it goes. The number of molecules of this enzyme is going to be limited, so it's called the rate-limiting enzyme. Often it's the first enzyme in the reaction sequence. So the first enzyme, enzyme A in this case, kind of sets the pace for the rest of the reaction to occur. In some pathways, the end product actually inhibits the rate-limiting enzyme, which is an example of negative feedback. So if we have enough of the product and we want to shut it off, then the product will actually go and bind to the original substrate so that the reaction turns off. Cofactors and coenzymes are things that make enzymes function better. Cofactors are non-protein substances that combine with the enzyme to actually activate it. Some of them help the enzyme bind to the substrate. Some of them help the active site fold into its proper conformation. Now, it can be a small organic molecule, an ion, or an element. Coenzymes are organic molecules that act as cofactors. Most of these are vitamins, which are essential to us that we have to actually get from our diet. Deficient or absent enzymes block metabolic pathways that they catalyze. So, for example, Hex A is a big enzyme that we need, and individuals that do not have Hex A have Tay-Sachs disease. So they can't catabolize that reaction. And what happens is lipids start to build up in their brains. Another example is PKU. There's missing and non-functional enzymes that are going to block the conversion of phenylalanine, which is an amino acid, into tyrosine. So what happens is excess phenylalanine enters the blood and will poison the brain. Now this can actually be treated with a special diet. You just have to avoid phenylalanine, basically. Energy itself is the capacity to change something or the ability to do work. Heat, light, sound, electrical energy, chemical energy, mechanical energy, these are all common forms of energy. The first law of thermodynamics states that energy cannot be created or destroyed. It can only change from one form to another. And cellular respiration is the process that transfers energy from the molecules and makes it available for cellular use. So this is how we generate ATP. Most metabolic reactions actually use chemical energy. So ATP, as I just said, this is generated from cellular respiration, actually carries energy in a form that the cell can use. So ATP is known as the currency of the cell because it's the energy of the cell. So ATP is adenine, which remember is a base, a nitrogenous base, ribose, which is a sugar, and then 3 phosphate groups attached to it. Now that third phosphate group is really unstable and it has a lot of energy in that bond. So if you break off that third phosphate, it can then attach to another molecule and give its energy with it. So it can fuel a reaction that could happen. And then you can attach that base again and you have your ATP back. So ATP and ADP, which is adenosine diphosphate, are often cycling in our body. So we have ATP with all of this energy built up, and then it will detach and energize another molecule, and you're left with ADP. Well, then after the reaction is catalyzed, we can form ATP again. And it just constantly goes back and forth between ATP and ADP. As I just said, ADP can be converted back into ATP by attaching that third phosphate group, which is called phosphorylation. Phosphorylation requires energy from respiration, but this ATP and ADP cycle back and forth between respiration and other energy utilizing reactions. So we're going to have this constant transfer of energy between reactions that create energy and then reactions that require energy. Many metabolic processes require the energy stored in ATP, and that energy is held in those chemical bonds, and then when the bonds are broken, the energy is released. Oxidation releases energy from glucose, and energy is then used to power respiration. In the cells, enzymes initiate oxidation by lowering the activation energy. Energy is actually transferred to ATP, but 40% is released as chemical energy and 60% is released as heat. So the majority of the energy is actually lost as heat. But this helps us maintain our body temperature, so it does serve a purpose. So respiration of glucose occurs in 3 reaction sequences: glycolysis, the citric acid cycle, and the electron transport chain, also called oxidative phosphorylation. So glycolysis is anaerobic, and it happens in the cytoplasm. The citric acid cycle and electron transport chain are both aerobic, and they happen inside the mitochondria. So we move from the cytoplasm into the mitochondria. So metabolic pathways are cycles. A product reacts to replenish the original substrate. So the citric acid cycle is an example. So you start with one substrate, and then an enzyme catalyzes it to the second substrate, another one to the third, and so on. So you have all these enzymes catalyzing each step of the cycle. Glycolysis, this is showing you, starts in the cytoplasm, and then the products of glycolysis will enter the mitochondria as long as oxygen is present. Because remember, glycolysis is anaerobic, so it doesn't require oxygen. If oxygen is present, then we will move on to the citric acid cycle. So cellular respiration requires a supply of glucose and oxygen. It produces carbon dioxide, water, ATP, which is a big thing, that's what we need, and then heat. There are aerobic reactions that require oxygen, and these make the most ATP. Anaerobic reactions do not require oxygen, but they do not make a lot of ATP, so they're not very effective in the long run. So glycolysis is the first reaction in the sequence of glucose breakdown. It's actually a series of 10 reactions. It breaks down the 6-carbon glucose molecule into 2 3-carbon pyruvic acid molecules. Happens in the cytosol. Anaerobic, remember, but it only yields 2 ATP molecules. So that's not a whole lot. So the 3 phases of glycolysis are the phosphorylation of the glucose, the splitting of the glucose into 2 3-carbon molecules, and then the production of NADH, ATP, and 2 molecules of pyruvic acid. Now, again, since this is anaerobic, it doesn't require oxygen, so it can happen in all organisms and it can happen really fast. But it's only going to generate 2 ATP molecules. So this would be the type of thing if you want to think about if you're going to sprint. You're going to sprint for 100 yards. You need a lot of energy quick. You don't really breathe when you're sprinting short distances, so you're not really getting that oxygen in. So glycolysis is good for that kind of situation. So here's the 3 phases in a little more detail. The first is phosphorylation. The glucose is going to be phosphorylated, and 2 ATP are actually used during this. Phase 2 is the splitting the 6-carbon molecule into 2 3-carbon molecules. And then phase 3, you're going to have 2 pyruvic acid molecules formed, 4 ATP formed, and 2 NADH formed. Now you say 4 ATP is formed, but remember you used 2 ATP in the first phase. So that's why you have a net gain of 2 ATP. If oxygen is present, the NADH and the hydrogen are going to deliver electrons to the electron transport chain. Oxygen is going to be the final electron acceptor. If oxygen is not present, there isn't an electron acceptor, so we shut down this reaction. No oxygen, it shuts down, and you keep having to go back to the glycolysis reactions in what we call fermentation. Lactic acid fermentation will occur if oxygen is absent, because what happens is the NADH and the hydrogen deliver the electrons and hydrogen back to pyruvic acid, which is going to form the lactic acid. Buildup of lactic acid is going to inhibit glycolysis and really lower the ATP production. As I said, glycolysis produces a lot less ATP than aerobic respiration, so you only have a net gain of 2 ATP per molecule. So if you want to sprint and that's it, this is fine. However, if you want to run a marathon or even a mile, you need to breathe. You need to get that oxygen in. So the rest of the aerobic reactions are going to involve oxygen. Pyruvic acid is going to enter the metabolic pathways, and we're going to have synthesis of acetyl coenzyme A occur. So what will happen is that pyruvic acid is going to bind with acetyl coenzyme A, and it's going to produce acetyl coenzyme A. So pyruvic acid moves from the cytosol to the mitochondria. The pyruvic acid is used to produce the acetyl coenzyme A. Basically what happens is the pyruvic acid binds with the coenzyme A, and the coenzyme A cuts off a carbon to leave you with acetyl coenzyme A. So the end products are carbon dioxide, water, and up to 36 ATP per glucose molecule. So that's a big difference. You have only 2 ATP from glycolysis, but if you continue on with aerobic respiration, you can get up to 36 ATP. So here's a summary of the citric acid cycle. For each acetyl coenzyme A, it's going to combine with oxaloacetic acid to produce citric acid, which is then going to be changed through a series of reactions, and the cycle is going to keep repeating itself as long as we have pyruvic acid and oxygen. 1 ATP is produced, and 8 hydrogen atoms are transferred to NAD+ and FAD. 2 carbon dioxide are produced as well. But remember, this is for each molecule that we have of pyruvic acid. So 2 go in. So the citric acid cycle actually turns twice for every glucose molecule, basically. NADH and FADH2 are going to carry the hydrogen and the high energy electrons to the electron transport chain. Now, the electron transport chain is just a series of enzyme complexes that are on the inner membrane of the mitochondria. So the energy from the electrons is going to transfer to the enzyme ATP synthase. ATP synthase, if you look at the name, it's an enzyme, and it's the enzyme that synthesizes ATP. So it's going to use that energy to catalyze the phosphorylation of ADP to ATP. Water is going to be formed, and oxygen is that final electron carrier. So with the electron transport chain, the electrons are basically transferred from complex to complex to complex until it gets to the ATP synthase. And then it's going to fuel the production of ATP from ADP. So here's an overview of the whole shebang. You have glycolysis, which is anaerobic in the cytosol. A 6-carbon glucose is cleaved down into 2 3-carbon pyruvic acid molecules. Then each pyruvic acid molecule is going to combine with coenzyme A. Coenzyme A is going to chop off a carbon and leave you with acetyl coenzyme A. Carbon dioxide is given off in the process. But remember, 2 pyruvic acid molecules go into the citric acid cycle. So the citric acid cycle actually turns twice per glucose molecule. And it's going to create 2 ATP as well. So we have 2 ATP from glycolysis. We have 2 ATP from the citric acid cycle. The big moneymaker, so to speak, is the electron transport chain. All of the electrons from glycolysis and from the citric acid cycle are transferred to the electron transport chain by NADH and FADH2. And then it's just a cascade of enzyme complexes that the electrons are going to be passed from complex to complex to complex until they get to the ATP synthase, which is an enzyme that synthesizes ATP. And then that's going to catalyze the conversion of ADP to ATP. And that will give off 26 to 28 ATP for a grand total of somewhere between 32 and 36 ATP, depending. Carbohydrates from the foods can either enter catabolic pathways for energy production or enter the anabolic pathways for storage. They can also react to form some of the amino acids. Excess glucose is usually converted into glycogen. Glycogen will be stored in the liver and muscle cells. Also, it can be converted into fat and stored in adipose tissue. This is just showing you how proteins, carbohydrates, and fats are all broken down in our bodies and where they can enter the pathway at. Then we have protein synthesis. Nucleic acids, deoxyribonucleic acid in particular, our DNA is the genetic material that basically tells us what proteins we will make and what we won't make. So enzymes, blood proteins, structural proteins, antibodies, cell membrane components, all of these proteins are going to be made according to the DNA instructions. Genetic information are just instructions that tell us how to use proteins, how to make them, and it's stored in the DNA sequence. A gene is a sequence of DNA that contains the information for one protein. And the genome is the complete set of the genetic information in a cell. An exome is a small portion of the genome that codes for proteins. And then we're going to express whichever proteins the DNA basically tells us to express. So gene expression is how we control what proteins are produced in each cell, how much of the protein is produced, and under what circumstances the proteins are going to be produced. Nucleotides are the building blocks of DNA. They are made of the 5-carbon deoxyribose sugar, a phosphate group, and a nitrogenous base. So adenine, cytosine, guanine, or thymine. Adenine, remember, is from ATP as well. The DNA resembles a twisted ladder. It's a double helix. So you have the 2 ladder legs, which are the sugar phosphate backbone, as we call it. And then the rungs of the ladder are those nitrogenous bases. So that's how they're connected. And then it spirals up in a double helix. The bases form 2 complementary strands, and they're linked together by hydrogen bonds. Remember, hydrogen bonds are those weak bonds between molecules, but they're strong enough to hold the DNA together and weak enough to let us open it up to get to a gene that we might need. The base pairing is C to G and A to T, so adenine binds with thymine and cytosine binds with guanine. This is just showing you DNA. So those dotted lines represent the hydrogen bonds that hold the 2 strands together. So as I said, it's a double helix, and each nucleotide has that sugar, which is deoxyribose, the phosphate, and a nitrogenous base. The hydrogen bonds hold those bases together, and they specifically pair with A to T, C to G, and this is known as complementary base pairing. DNA wraps around the histone proteins to form chromosomes. So when the cell is not actively dividing, the DNA is kind of stringy looking, and it's called chromatin. As the cell gets ready to divide, the DNA wraps itself around those histone proteins and gets tighter and tighter and tighter until it's condensed down into a chromosome. The human genome contains about 3.2 billion bits of information. And DNA profiling and fingerprinting actually compares the most variable parts of the genome among individuals. So it's going to be used to identify human remains at crime scenes or even during natural disasters. When we have earthquakes and tsunamis and all of these things happen, unfortunately, DNA has to be used to identify some people. It can also check for family relationships, especially paternity tests. You are not the father. Or it can establish innocence in criminal cases such as rape cases. Hundreds of prisoners have actually been freed because of DNA profiling. They were wrongfully convicted when they didn't have the technology that we have today. And now that we have the technology, things are being retested and prisoners are being set free. So DNA replication occurs when DNA has to copy itself. So an exact molecule of DNA is going to be produced, and this occurs during interphase. Hydrogen bonds are going to break between the base pairs. The strands are going to unwind and separate. The new nucleotides are going to pair with exposed bases under the direction of DNA polymerase. So that's an enzyme. And then other enzymes are going to connect the new sugar phosphate backbone back together. So the first thing that has to happen is the hydrogen bonds are broken. The strands unwind and separate. DNA polymerase comes in and brings the complementary base pairs, and then it winds back up. So here's a picture showing you, and this happens during the S phase of interphase in mitosis and meiosis. So the genetic information also stores the sequence of amino acids to make proteins. The triplet code is the 3 nucleotides that represent an amino acid, or it can signal the beginning or the end of a protein. The sequence of bases in a gene is going to determine what amino acid is there. And DNA, of course, stores the master copy of the genetic code and stays inside the nucleus. So protein synthesis occurs in the cytoplasm, but the RNA is copied and transfers the information from the DNA. So the RNA is made inside the nucleus, and then it leaves the nucleus and goes to the cytoplasm to direct the synthesis of the protein. RNA molecules are single-stranded. They have the sugar ribose instead of deoxyribose. They still have the phosphate group. And their bases, they have adenine, guanine, and cytosine, just like DNA does, but they have uracil instead of thymine. So the complementary base pairing in RNA is C to G still, but it's A to U. It's a lot shorter than DNA, and transcription is the process of copying that DNA sequence onto an RNA sequence. So messenger RNA carries the message. It carries the genetic code from the DNA to the ribosome. RNA polymerase catalyzes the formation of messenger RNA from DNA. So for replication, just to recap real quick, replication is DNA to DNA. DNA polymerase catalyzes that reaction. So the hydrogen bonds will be broken, the DNA will unwind, DNA polymerase comes in and matches the base pairs according to complementary base pairing, and then it winds back up. When we need RNA polymerase is going to bring the new bases and match them according to the base codes. So RNA polymerase is going to catalyze the formation of messenger RNA. So RNA polymerase recognizes which DNA strand needs to be copied. That section of DNA is going to unwind and expose the gene for that particular protein. Because keep in mind, we have thousands of genes on one strand of DNA. So we only have to unwind a section for this, whatever the gene we want coded is. The complementary messenger RNA nucleotides will pair with the DNA bases. Uracil, remember, instead of thymine. A termination signal is going to indicate that's where the gene ends. And then the new messenger RNA strand is going to be released. And then the DNA winds back up. The messenger RNA leaves the nucleus through a nuclear pore and is going to attach to a ribosome in the cytoplasm. So this is just showing you RNA bases that match DNA. Once the RNA combines with the ribosome, now we're in the process of translation. Each amino acid sequence is going to be specified by 3 bases called codons. Protein synthesis happens in the cytoplasm because, remember, the messenger RNA left the nucleus to bind to the ribosome. At the ribosome, it's going to read the messenger RNA. So think about translation. If you translate for somebody, you're kind of reading it to them. The ribosome does the same thing. It translates that piece of messenger RNA into the sequence of amino acid that will become the protein. So here's an overview of protein synthesis. So just to recap, replication is DNA making DNA, copying itself. DNA polymerase runs that show. Transcription is messenger RNA being made from DNA. RNA polymerase runs that show. And then translation happens in the cytoplasm when the messenger RNA leaves the nucleus through a nuclear pore and binds to the ribosome. Then the ribosome will translate that messenger RNA into a protein. So it's reading it in a nutshell. It reads every 3 bases, which is called a codon, and then transfer RNA, which is another type of RNA, will bring the correct amino acid to the ribosome. So protein synthesis requires amino acids being added in a proper sequence. Transfer RNA aligns the amino acids during protein synthesis. It's going to bind to its amino acid, transport it to the ribosome, and then bind to the messenger RNA according to the sequence. It adds an amino acid and it will leave. Each transfer RNA contains that 3 nucleotide bases called the anticodon, which is going to be complementary to the codon on the messenger RNA strand. So as the ribosome moves down the messenger RNA, each transfer RNA will bring its amino acid, and the polypeptide chain is going to keep growing. Now there are 20 types of amino acids. We have 64 possible codons. Most codons correspond to amino acids. 1 codon will code for each amino acid, but sometimes there could be a few codons for a particular amino acid. We do have a few things that are set though. AUG codes for methionine and also is the initiation codon, so it signals the start of the protein. We have 3 stop codons, which signal the end of the protein, and then each messenger RNA codon that codes for amino acids has a corresponding transfer RNA anticodon. So each codon has an anticodon. So this is the triplet code, basically, the genetic code. So 3 bases will represent the particular amino acid. Notice that the 3 stop codons are up in the top right-hand side, UAA, UAG, and UGA. If the ribosome gets to that point and the messenger RNA has a UAA, that tells the transfer RNA to stop and there are no more amino acids that need to come. AUG is the start codon, so methionine. Just remember that school starts in August. So school starts in August, AUG is the start codon. And then notice some of these amino acids have 4 codons, like leucine, or leucine actually has 6 codons. Histidine has 2, arginine has 4, lysine only has 2, so you kind of get the basic gist. There could be quite a few codons for one amino acid. This is just showing you the part of the messenger RNA molecule, showing the codons and then the amino acids they represent. So protein synthesis occurs on the ribosomes. Messenger RNA is the template. Messenger RNA was transcribed from DNA. And then transfer RNA brings the amino acids to the ribosome, binds to the messenger RNA, and adds its amino acid to the growing polypeptide chain. Ribosomes are composed of ribosomal RNA. They have two unequal subunits, a large one and a small one. And what will happen is the messenger RNA binds to the small unit, which locks down the large subunit. And that's going to be able to push the messenger RNA through the ribosome. Amino acids will be joined by peptide bonds. And then when the ribosome reaches the stop codon, the protein will be released. This is just summarizing transcription in the nucleus and translation in the cytoplasm. 99% of the human genome sequences are the same among all people. That 0.1% of the genome that varies includes DNA sequences that can affect health, appearances, and other ones that have no observable effects. So needless to say, we have a lot of things in common with each other, but as you know, we also have a lot of differences. Mutations are changes in DNA sequences. They occur if a base is going to be changed, added, or deleted. Now, they can be spontaneous, which means that an unstable base is just inserted mistakenly, or they can be induced due to exposure to mutagens. Mutagens are just things that cause mutations, like chemicals or radiation. Some mutations are not harmful at all. Many mutations affect health, though. They can change the amino acid sequence, which results in a non-functional protein. Duchenne muscular dystrophy, for example, results in a mutation in the gene coding for dystrophin. Now, dystrophin is the protein that kind of stabilizes the muscle membrane. So without that stabilization, when contraction occurs, the muscle cells collapse. So this results in severe muscle weakness. Very rarely a mutation can actually provide an advantage to health. There is a mutation that protects some people against HIV. The receptor that the virus binds to is incomplete, so the virus can't bind to the cells. So that's one of the rare exceptions. DNA does have a repair mechanism. If a nucleotide is mismatched, a repair enzyme can go out and fix it. 2 to 4 codons often specify the same amino acid, though. So some mutations result in the production of the same amino acid, and it doesn't affect the protein. We also have 2 chromosomes. So if one copy happens to be mutated, the other copy may provide enough of the gene's normal function to maintain our health. So even though we have all of these dangerous things that can happen, because the genetic code is the way it is, we have some protection against the bad stuff.